Good morning, friends. So, after discussing about the gene interaction in last lecture, now it's a time to discuss about another modified Mendelian inheritance, that is epistasis. So, let us begin with epistasis. So, what is epistasis? It is a Greek word which is made up of two different words, that is epi and stand. So the meaning of these two words is standing above. Epi means above and stand means stand. So that means something is standing above. So that is the literal meaning of epistasis. So in case of epistasis, the expression of one gene pair either mask or modify the effect of or expression of another gene pair. That is the basic concept of epistasis. In other words, we can say that when the allele of one gene masks the phenotypic effect of alleles of another gene that is present at different locus, this phenomena is known as epistasis. So this is the definition of epistasis. So when I am telling that the effect of one gene at one locus is getting suppressed by another gene present at another locus, that means there are two components: the allele or gene which effect is being masked and allele or gene which is masking the effect. So related to this, there are two terms, the epistatic gene and hypostatic gene. So by name you can understand the one whose effect is being masked is called as hypostatic gene, and the another who has masked the effect of another gene is called as epistatic gene. So the epistatic gene may be dominant or recessive in their effect. So now let us start with the different kinds of epistasis. So first of all, recessive epistasis. So recessive epistasis is showing the dihybrid F2 generation ratio, phenotypic ratio, as 9:3:4. So you have to remember these ratios in terms of normal Mendelian dihybrid F2 ratio. So we know that the normal dihybrid Mendelian F2 ratio is 9:3:3:1, which will be modified to 9:3:4 in case of recessive epistasis. The example of recessive epistasis, one thing is that I have tried to include more and more example from animal side, but in many cases I failed to get or collect the examples from animal side. So there I have given example from plant side. So here in case of recessive epistasis, the example is pigmentation in mice, that is the skin coat color of mice. So the wild type coat color is agouti, which is dominant in their expression, and another color is, and the dominant to which color? The solid color, for that is small a. However, this coat color is being controlled by a separate gene, that is C gene, which is necessary for pigment production. And now the mouse with a recessive C allele at this locus is unable to produce pigment and is albino regardless whatever gene is present or whatever allele is present at a locus. That means the capital C, the C, dominant C is necessary for production of color. So here you can see when agouti mice is mated with agouti mice. So in F2 generation you can see when there is presence of dominant A That means कि agouti color is capital A is dominant over small a small a. So when there is recessive C at another loci, so in that case there will be no color development. On all other case there will be agouti color development, but when there is no dominance at A locus and there is dominance at C locus, in that case the grey color mice will be or black color mice will be produced. So finally you can see the agouti, black and albino will be produced in the ratio of 9:3:4. So recessive alleles from one locus block the expression of both dominant as well as recessive allele at the other locus. So you can practice it by your own. You just take a paper and try to form Punnett square with the given genetic composition. Then you will find all your answers, and it's better to apply the probability rule that we had discussed when I was discussing you about the normal Mendelian dihybrid ratio. The product law of probability will help you to predict the genotypic and phenotypic ratio in case of epistasis also, or in any case. So this was about the recessive epistasis. The second one is dominant epistasis. Sorry, this is another example of recessive epistasis, that is coat color in Labrador. So here again you can see that capital E gene is very essential for extending the color produced by B gene. So here you can see that if capital B and capital E both are present, the animal will be black in color. If there is no capital B, that means there is no dominance at B gene, but E extender gene is present, then it will be of chocolate color. But when the extender gene is not present in dominant position, in that case the color will be yellow color. So this is the example in case of dog for recessive gene, recessive epistasis. Now coming to the dominant epistasis. So in dominant epistasis, the modified Mendelian ratio will become 12:3:1. So this is all I am telling you about the phenotypic ratio. Genotypic ratio will remain same as we had seen in the normal Mendelian dihybrid cross. So here what we have to observe, that dominant allele at one gene locus masks the effect or action of alleles at other locus. So there recessive gene was blocking the action, but here dominant allele will mask the action at another gene locus. So here in this example you can see how the things are changing. So at one locus, this you can observe that at A locus, so A locus will mask the effect present at B locus. When A is in dominant position, it will control the B locus also. So that's it called as dominant epistasis. So here the example is coat color in dogs. So here you can see that the B locus and I locus, two locus are discussed. Small b, small I is brown color, and capital B, capital I is white color. That means the I is inhibitory gene. So I, whenever dominant I will be present, dominant allele will be present at I locus, it will mask the effect of B locus. So here you can see when these two dogs are crossed, the resultant dog is again white because there is dominance at I locus. Although there is dominance at B locus also, but since I is epistatic gene, it will mask the effect of B allele. So if you cross this white color, that is F1 or intersemate, if you intersemate, then you will get the ratio of 12:3:1. Why? Because the dominant allele at I locus will have the control. This is also known as supplementary gene action. Supplementary gene action. So here you can see, you just go through this Punnett square, you try to do it in your notebook, then you will get clear cut idea, and always remember the product law of probability. So coming to the next example or next epistasis, this is one more example that is in the plant, that is fruit color in squares. This again example of dominant epistasis. Here you can see again that when capital A small a, capital B small b is crossed with capital A small a and capital B small b, similar in the case of coat color in dogs. Here when B gene is present in dominant form, it will inhibit the development of color. But when B gene is recessive and A gene is dominant, either homozygous or heterozygous, it will have different color. Here you can see it is yellow color. When both are recessive, then it will have different color, that is green color. So this example here you can clearly appreciate the action of dominant genes. Now coming to the duplicate dominant epistasis. So as the name suggests, duplicate dominant means the dominant allele at both gene loci produce the same phenotype. So the dominant alleles at both the loci, it will produce the same color. This is also called as supplementary gene action. Supplementary gene action. So here you can see the example is feathered shank, featherness or non-featherness of shank in poultry. So in a cross between black Langshan, that is having feathered shank, and buff rocks, these are the different strain of poultry that is having unfeathered shank, the shank of F1 generation will be all feathered. Here you can see the two genes are there. What are those two genes? The F and S, two alleles at different locus are involved. So when there is dominance at both the loci, it will have feathered shank. When there is no dominance at both the loci, it will have unfeathered shank. So when it is crossed, there will be feathered shank. And in the F2 generation, you can see the ratio is 15:1. That suggests that when there is all recessive, that means recessive at both the loci, then it will have different phenotype, that is unfeathered phenotype. Otherwise, in rest of the cases, dominant allele present at either or both the loci will have similar type of phenotype, that is feathered phenotype. So this will result in the ratio of 15:1, phenotypic ratio of 15:1. Now, the another epistasis is duplicate recessive epistasis. Duplicate recessive epistasis. The similar in case of duplicate dominant epistasis. Here, in duplicate recessive epistasis, the recessive homozygous at either or both the loci will mask the expression of colored allele and result in white phenotype. So here I could not find the example from animal side, so I have taken it from the plant, that is sweet pea. Here you can see the white variety, that is capital C, small p, and another white variety small c, capital P. Here you can see that recessive alleles at different loci. Both are phenotypically white, but when they crossed, there will be formation of heterozygote individual, and it will have the purple color flower. So when these plants will be intersemated, the resultant plants, you can see it will be in the ratio of 9:7. Why 9:7? Because recessive alleles at either loci, or both the loci will have same phenotype, and rest of the genetic composition will have different phenotype. So the color of sweet pea flower is controlled by two genes which are epistatic to each other and show complementation. That is why it is known as complementary gene action. So here you can see that colorless precursor, there will be presence of colorless precursor, but when there will be C enzyme, that is dominant C, it will produce colorless intermediate. Intermediate will be produced, that is again colorless, but when capital P, that is enzyme P is present, that is dominance at P position, then it will produce purple pigment, that will be colored pigment. So this is the mechanism behind the duplicate recessive epistasis, and it will result in the ratio of 9:1. So again you draw Punnett square in your notebook and get acquainted how this ratio is getting changed from 9:3:1 to 9:7. So this was about the duplicate recessive epistasis. Now the another one is duplicate dominant epistasis with cumulative effect. So the only difference from duplicate dominant epistasis is that when dominant alleles are present at both the loci, they will have different distinct phenotype. So here you can see dominant homozygous or heterozygous at either loci, but not at both loci, will produce the same phenotype. So when there is dominance at both the loci, they will have distinct phenotype. So this is the example is in Duroc Jersey breed. Here you can see this is dominant at S locus and dominance at R locus. So after crossing, it will produce the brown pig that will be dominant at both the loci, although heterozygous but dominant at both loci. Now when they will be interseminated, they will produce animals in the ratio of 9:6:1, which indicates that dominant at both the loci will have different phenotype, that will have the brown phenotype, otherwise all the other animals will have sandy phenotype, and when there is no dominance at both the loci, then it will have white phenotype. So this is about the duplicate dominant epistasis. It is duplicate dominant epistasis with cumulative effect. It is similar to duplicate epistasis but with cumulative effect, which tells that dominant at both the loci will have different phenotype. Now the next and probably the last one is the dominant-recessive epistasis. Dominant-recessive epistasis. What it tells, the dominant allele at one gene locus in homozygous or heterozygous condition and the homozygous recessive allele at other gene locus produce same phenotype. Dominant and recessive. So what it is telling, that dominant alleles either in homozygous or heterozygous at one locus and recessive alleles or in a homozygous condition at another locus will produce same phenotype. So here again the phenotypic expression or phenotypic classes will be limited to two only, because dominant either in homozygous or heterozygous and recessive in homozygous condition will have same phenotype. So first two and last one will be combined together, and this will give the ratio of 13:3. At last I will tell you how to remember these ratios. So now this type of inheritance is called as inhibitory factor inheritance. So the example is the color development in poultry. So here you can see the white leghorn feather color. White leghorn, it is controlled by two genes I and C. Again white leghorn. Now when they cross together, they are heterozygous, and when they were interseminated, the phenotypic ratio is coming in form of 13:3. So all 13 white and 3 are colored. The 3 which are colored is having they are heterozygous at the second loci. Because either homozygous or heterozygous dominant at first loci and recessive at second loci will have same phenotypic expression. So this was about the dominant recessive epistasis. Now I think I have done with the epistasis. Now the how to remember. So first of all, I will write the ratio of normal Mendelian phenotypic ratio, dihybrid phenotypic ratio. It is 9:3:3:1. So when there is dominant epistasis, you have to combine these two dominant classes. So the ratio will be 12:3:1. When it is recessive epistasis, you have to combine last two recessive phenotypic classes, which is expressed due to recessive allele. So the changed phenotypic class will be 9:3:4. 9:3:4. Now, okay. Again, now you can see, again I am drawing, okay. No, it is not changed. Actually, I am trying to change the color. Okay, done. So now we can see 9:3:3:1. In case of duplicate dominant epistasis. So duplicate dominance epistasis means dominance at either loci. So what will happen? All the dominant phenotype will be combined together. That is 15:1. 9:3:12 or 3:15. 15:1. Similarly, in case of duplicate recessive epistasis, all these three will combine. So it will become 9:7. Again, if it is dominant recessive epistasis or with cumulative effect. So with cumulative effect, the effect at both loci means dominant at both loci will have distinct phenotype. So in that case, the ratio will be changed to 9:6:1 because the probably 9:6:1. I will just check and tell. Yes, with cumulative effect, 9:6:1. So what will happen? This intermediate phenotype will combine together because dominance at both loci will have different phenotype. So this is how we can remember the different epistatic ratio. And the important thing is that you just draw all the F2 F2 crosses on your notebook, and then you try to correlate it with the normal F2 dihybrid cross ratio, and then you will get clear-cut idea about the epistasis. There are many other epistasis also present in the nature, but for your course, I think this much is enough. Thank you very much for your patience listening.